Good afternoon, everyone, and on behalf of the Biochemical Society and Portland Press, I'm pleased to welcome you to this today's webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. So please see our website for more details. I'm Professor Rachel Simmonds. I'm Professor of Immunopathogenesis and incoming Head of Department of Microbial Sciences at the University of Surrey. My research uses cell and molecular biology to investigate the pathogenesis of Buruli ulcer caused by Mycobacterium ulcerans, and in particular the function of its exotoxin, mycolactone. I've been involved with the Biochemical Society for a long time, and I'm here as I'm currently a member of the Biochemical Society's Awards Committee, and I've been doing that since 2020. In this webinar, we're celebrating the 60th anniversary of the Colworth Medal, kindly sponsored by Unilever. The Colworth Medal is a prestigious annual award presented for outstanding research by an early career biochemist of any nationality who's carried out the majority of their research in the UK or the Republic of Ireland. In this series, we'll be bringing together some of our past winners from across the decades to discuss their careers, achievements, and to give advice to ECRs and people at all stages of their careers. Today, I'm delighted to be joined by Professor Sir Mike Ferguson, Professor Simon Boulton, Professor Helen Walden, and Professor David Grainger. Before I start the introductions, I'd like to remind everyone that questions are encouraged throughout the webinar. So please type the questions into the box as shown in the image on the screen, and we'll try and get through as many as time allows. So I think we'll start off with a round of introductions, if that's all right, panel. So I'll start with Mike Ferguson, who was awarded the Colworth Medal in 1991, and next year will receive the Morton Lecture Prize. Please go ahead, Mike.
Thanks, Rachel. So yeah, Mike Ferguson. I'm Regius Professor of Life Sciences at the University of Dundee, where I've been for 35 years now. I'm a parasitologist and glycobiologist, and with a great interest in drug discovery and trying to translate discoveries into new therapeutics for neglected tropical disease.
Lovely. Thanks, Mike. And then next, so I'm going through these in order of award year. So next is Simon Boulton, who was awarded the Colworth Medal in 2006.
Thank you, Rachel. Thank you for the invitation to be here. So yeah, I'm a molecular biologist and geneticist. I'm currently a principal group leader at the Francis Crick Institute in London. I'm also co-founder and VP Science Strategy of a biotech company that we span out called Artius Pharma, and I'm also director of a radiation initiative set up by Cancer Research UK, which is called RadNet, and I look after cross-institutional collaborations within the London area.
Lovely, thanks, Simon. And next is Helen Walden, who received the Colworth Medal in 2015.
So hi there, I'm Helen Walden. I am a Professor of Structural Biology and the Head of the School of Molecular Biosciences at the University of Glasgow. I've only been in Glasgow for about 6 years. This is the third time I've started my lab, and I am interested in understanding molecular mechanisms of cell signalling, primarily by ubiquitination.
Great, thanks, Helen. And last but not least, we have David Grainger, who won the Colworth Medal in 2016.
Hi, thanks, Rachel. So my name's David Grainger. I'm at the University of Birmingham. I've been here on and off since first coming as an undergraduate in 1996. So I've left several times and always managed to find my way back. My interests are molecular microbiology, particularly with respect to the regulation of genes.
Great. Okay, well, it's really nice to, some of you I've met before, but it's really nice to meet the others of you today. So I thought we'd start off by talking about your career pathways, because we know there's lots of ways to career success. And so if you could each talk us through your career pathway And if you could describe what stage of your career you won the Colworth Medal at, and perhaps if that kind of helped your career in any way, if we could, yeah, if you could focus on that as you talk through it, that would be great. So we'll start with Simon. As I said, I'm going to be sort of going around my screen, if that's okay.
Yeah, thank you, Rachel. So I grew up in Leeds. We'll mention the football team. But I grew up there and I became fascinated in science. I had a couple of teachers who were really inspirational, and I ended up going to Edinburgh to do my first degree and studied molecular biology. I actually went to do chemistry, and I was very fortunate to be able to change after the first year. And I think that was quite a difficult decision, but I think it certainly was the right decision for me. I was fascinated with the emergence of genome engineering, those kind of things. I mean, it's not CRISPR at this point. It was, you know, restriction enzymes and what have you, and picking things back together. And I ended up going to do a PhD in Cambridge with Steve Jackson, and that was very important for me. Again, the environment was wonderful in Cambridge, and I think the message I would have to early career scientists is to find a really strong collaborative environment surrounded by really exceptional people, because I think it really brings your level up. So I then did postdocs at Harvard Medical School, 2 at some other cancer hospitals, Mass General and Dana-Farber, and then came back to the UK in 2002. So I've had my lab just over 20 years now, and we've used a variety of different model systems. C. elegans, yeast was where I started, Drosophila, mammalian cells, we use mouse models, and lots of different multidisciplinary approaches, from biochemistry, single molecule approaches now, gene editing, CRISPR screening, those kind of things. So that's been something that the lab has evolved into over the time. I won the Colworth Medal 2006, so it was 4 years into running a lab, so I was still kind of an early career group leader, a junior group leader, so to speak. And I think for me, I mean, it was a very big deal at the time, because it gave the lab a lot of prestige. We got some publicity around it, and I think that opened a few doors for us. You know, I say us, me, I'm referring to the lab, and I think for me it was very important for my career trajectory. It was the first award that I received as a group leader, so it was very special. And there's, yeah, as I mentioned, I think in my introduction, I was very fortunate to work with Steve Jackson in Cambridge. And for those of you who know Steve, he was quite a pioneer. He set up the first DNA repair company, which was called Kudos, and they developed the PARP inhibitor olaparib that was subsequently acquired by AstraZeneca. And that line to translation, commercialization, kind of certainly had an influence on me. And you know, I was always had an eye on potential opportunities for translation. So you know, when we made some discoveries in, I think it was about 2010, and then it took us another number 5 or 6 years before we could fully realize the potential of this. So you know, we raised money, we started Artios, and we're now in phase 2 clinical trials with several different small molecule inhibitors that we've developed against DNA repair molecules. So it's really exciting. So I'm very fortunate that, you know, during my career I've had the opportunity to learn about not just academia but industry developments. So yeah, I kind of, a bit like Mike, have bridged the two sides of academia and industry, and so I feel very privileged for that.
Great, thank you very much. And I think there's some themes there that we might return to later on as we go on. I mean, it feels like a good time to go to Mike because of that link with drug development. Yeah, would you like to answer the same question, Mike? Thanks.
Yeah, okay, thanks. Yeah, I did I got interested in biochemistry and studied it at Manchester, and then did a PhD in London, and then a postdoc at Rockefeller University, and then another one at Oxford University. And the subject area I got into, molecular parasitology and carbohydrate structure determination and so on, was a bit of an accident. I was absolutely determined when I went to interview for PhDs that I wanted to do something in cancer research. But when one of the interviews happened to be in parasitology, and then I learned just how neglected that whole area is in terms of therapeutics. So that's what swung me into that, and I haven't left that field since. And I took a lectureship at the University of Dundee in 1988, and I got the Colworth in 1991. So I was just 3 years into my first faculty position. And again, as Simon said, it was a very, very useful thing, because obviously there's a lot of kudos that goes with it, and so I really appreciated the exposure that it gave me to, particularly to the UK science community, but broader than that. So it was really, really useful. I guess my scientific career has gone from structure determination, did the first GPI membrane anchor structures, for example, but those are only structures are only roadmaps, in my opinion, for understanding biosynthetic pathways. The biosynthetic pathways took me to synthetic organic chemistry, because if you're working on a new pathway, there's nothing in the catalogs that you can buy, you have to make your own substrates. And that led me into really thinking seriously about drug discovery. In 2006, we set up a drug discovery unit in Dundee, which has grown to being quite substantial now. We've got 4 drugs in the clinic as we speak, 3 in parasitology, 1 in cancer, interestingly, in leukemia. And actually what I'm obsessed with at the moment is general commercialization of intellectual property from my university, but keeping the companies that come from it in Dundee, which is an incredibly economically depressed region. Up till now, we've been spinning out very high growth, highly invested companies, but they've ended up as part of a leveling down agenda in Oxford and Cambridge.
Right, yes.
I'm determined to stop that from happening. So that's my current obsession. And the other thing that I've done in my career, I guess, is served on some boards, the board of Wellcome Trust for 10 years. That was a fantastic privilege. And also Medicines for Malaria Venture, and most recently the UK Biobank. I'll stop there.
Lovely, thanks, Mike. Yeah, really interesting to hear those links there. Okay, Helen, let's move on to you next, if that's okay. If you could summarise your career in the same way. Thanks so much.
Thanks very much. So, so I think the theme with me is a bit of a nomadic experience. So I grew up in Germany and Cyprus and didn't move to the UK till I was 17. And I don't remember any teachers being particularly inspiring. I just quite liked science. So I went to the University of Bath to study biochemistry. And it was there, they had a sandwich course where you got to do 2 sets of 6 months placements. And my first placement was in a very nice place, but it was basically a diagnostic kits manufacturer. So you just did the same experiment all day, every day, and it was very dull. And I thought, oh, I don't want to do this for the rest of my days. And so in my following year, I got to go to the Weizmann Institute of Science in Israel and have a whole project. And I still remember coming back saying to my dad when he picked me up from the airport, I know what I want to do, I know what I want to do, I want to do research. So I ended up moving to the University of St Andrews to do my PhD with Gary Taylor, and that sort of ignited my love of Scotland. And then when I completed that, I went to St Jude's Children's Research Hospital in Memphis, and that's where I did my postdoc. I moved back to the UK, to London in 2005, which is when I started my group at Cancer Research UK. In fact, Simon and I were colleagues across different sites. And perhaps what we'll come back to later is that after getting tenure there, I was told I wasn't good enough to go to the Crick Institute. And that then propelled me into the next phase of, well, where do I want to carry out my studies next? So I moved to the University of Dundee, where Mike Ferguson was indeed a colleague there. And that was a really lovely 4 years that I spent in Dundee. And then for various personal and other reasons, decided that the University of Glasgow would be my next path forward. So that's the whistle-stop tour of Helen's career so far.
Lovely. And how about the Colworth Medal? What stage of your career were you at when you were
I was just trying to think about this. So I think one of the reasons why it meant so much to me is because having just had this fairly unpleasant thing happen with the Crick, I was then nominated for the Colworth that year and got it the following. And so it was really a nice way of sort of bandaging up some wounds. And it's certainly, I remember a very tangible couple of years later when applying for a big European Research Council grant. One of the reviewers went back through the illustrious list of Colworth winners and said, without question, every single one of them was a leader in their field. Therefore, you could bank on Helen, sort of thing. And that was really tangible. So for me, it was just, it was really nice. It was great recognition for, you know, we were doing hard stuff that took a long time to do, right? So it was in that sort of short-termism you can sometimes get in science. I think the Colworth Medal, although it came quite late, I'd been running my group for 9 years by the time I was told I'd got it. I think it was a recognition of the sort of doggedness and determination we were putting into our work.
Yeah, wonderful. And I think, yeah, that's an important message, isn't it? That there's great research outside of the Golden Triangle. And I think sometimes early career researchers can get quite sort of linkered into sort of thinking certain places are, I don't know, you're more likely to have a successful career at one place rather than another. Yeah. Okay, so last but not least, David, sorry to bring you in at this point now. And yeah, if you could answer the same question, please, that would be great.
Yeah, so I think my kind of career route is a little bit different in that at one point I took a deviation away from research. So I first came to Birmingham, as I mentioned earlier, in 1996 as an undergraduate. And I remember the first year, it just seemed a lot of hard work. You know, I was just slogging through all this stuff. But it was in the second year, and I'll maybe talk about it later. But it was the second year that I really got turned on to gene regulation in particular, and that became the thing that I was fascinated in. But when I got to the end of my undergraduate degree, I really wasn't quite sure what to do. So I took a job in industry, which I didn't stay in for very long. I actually trained as a teacher. And when I trained as a teacher, all that any of the other teachers said to me was, don't do it, which was a little bit off-putting. And I remember in particular, we went to a day in Telford, and we've got this super teacher, apparently a super teacher come to speak to us, and she said, the really smart people, and actually I don't agree with what she said, but the really smart people don't become teachers, they become scientists. And I thought, well, if that's the opinion of another teacher, then I think that's what I'll do. So I think that same evening, I got back in touch with some people at Birmingham and said, you know, could I come and do a PhD? So that's what I did. So I finished the teacher training, came back to Birmingham to do a PhD, loved pretty much every minute of it. So by the time I got to the end of the PhD, I knew exactly what I wanted to do. I stayed in Birmingham for a short time as a postdoc, went to the University of Warwick in 2008 with a Wellcome Trust Fellowship, and then never really expected to come back to Birmingham, but I did. I actually came back 3 years later, and I guess it was a few years after coming back to Birmingham. So yeah, so a few years after coming back to Birmingham, that's when I got the Colworth Medal. So it was kind of in between coming off the independent fellowship and being promoted to professor. It was in that interim period that I got the Colworth Medal. And it kind of really opened my eyes to, you know, lots of new areas, lots of new people. Coming to the Biochemical Society to sit on panels, that sort of thing was fantastic. And so I mentioned we're interested in gene regulation. So to start with, that was very much gene regulation in E. coli, looking at a very well-known model system. And then over the years, we've kind of diversified into some nasty pathogens and things like that. So at the moment, I'm getting interested in transposons for the first time.
Oh, fantastic. Great, thanks very much. I think that's really illuminating, isn't it? That there are a real range of different sorts of careers here that we've got some. So Mike, you stayed at the same place for a long time. You found a real home that's been successful, but yes, there's no real one recipe for success. So a couple of you talked about your specific teachers, or there not being specific teachers that really kind of were inspirational to you as you were thinking about pursuing a career in the molecular biosciences. I wonder, Mike or David, does either of you have a particular teacher or experience in mind that kind of, I mean, David, you sort of touched on that, didn't you, in terms of other teachers encouraging you to think in another direction. So perhaps, Mike, is there an experience you could talk to that kind of drove your career aspirations?
Yeah, I was always interested in science from being a very small boy. You know, from the age of 5 or 6, always really interested in anything sciencey and technologically. And I always used to love, you know, things with big dials and levers and buttons on it. And of course, the sad thing was, by the time I became a scientist, the microprocessors had been invented and everything was touchpads. But that was the only disappointment in life. But I had a great school, and I had a great physics teacher and chemistry teacher, Mr. Wise and Mr. Harris, and they were fantastic. And they used to do things like give me the keys to the physics labs of an evening and let me go and use the oscilloscopes and signal generators and anything else I wanted, unsupervised. Wow. It just wouldn't happen today, right?
No,
but that was so stimulating to be able to go and experiment unfettered at the age of 17. And I would say that had a big effect on me. Even though I went into bioscience rather than physics, it was a fantastic experience. I'm immensely grateful to those teachers. Yeah, thanks.
Lovely. Oh, great. Thank you. Well, let's move on to talk about how mentors, collaborators, support networks, and so forth have helped support your career as you've progressed through your career. And let's start with Helen this time, shall we? And then I'll move around again. Thanks.
Okay, thanks. Yeah, so I thought about this. I think that one of the advices I would give to early career researchers who perhaps are in a different demographic is that having a support network of people to remind you people who understand what it is you're trying to do and can help you sort of keep the faith when there are others knocking it. And I do think science to a certain extent favors extroverts. And I do think that it's very important to go to meetings, talk about your work, don't expect it to speak for itself. You do actually need to tell people what it is you're doing. And certainly the nomination for the Colworth came from somebody I'd met at a conference who'd seen some of our work, and then he got in touch with me. And so I think that there are mentors. I mean, I have mentors everywhere. I seek them out and I find and I do a lot of it in return. I think it's a really important, it's a human pursuit in the end, science is.
Oh, lovely, great, thanks, Helen. That's great to hear. Yeah, those kind of getting stuck in at conferences, I think, is quite important. It's one of the main reasons I quite like going to conferences on my own. It makes me less lazy. Okay, let's ask Simon the same question then, please.
Yeah, I completely agree with Helen. I think having supporting colleagues is really important for your, for your well-being. Science is very difficult, as I'm sure there people are realizing. Right, most of what we do doesn't work for whatever reason, but it's that 1 or 2% that does work that reveals something, transformative that we get so excited about. So as I mentioned, so Steve Jackson was a big influence on me in my career. But I think when I moved back from the US to Clare Hall, so I was at Clare Hall, Helen was at Lincoln's Inn Fields as part of the London Research Institute. So I was very fortunate to have colleagues who were working on related topics. So we were all working on sort of genome stability, DNA repair, DNA replication. And so it was a very small community of people who supported each other. And I think I was incredibly naive when I first arrived. I had no idea what this place was. You know, Tim Hunt had just won the Nobel Prize, I think, just after I had arrived. So I realized that, you know, the kind of science that was going on there was pretty special. But I think for me, you know, I've had some very special people who've kind of watched over me. I mean, one in particular, Steve West. I mean, he gave me an opportunity very early on when I started my group. We discovered BRCA2 in C. elegans, and this was quite a transformation for us because, for those of you who know, so this is one of the main breast and ovarian cancer tumor suppressor genes. It's an enormous protein. It's 3,418 amino acids in humans, and yet we found a C. elegans equivalent that was 1/10 of the size. So it was tiny, and what this we were able to really refine and understand some of the real basics. There's a lot of conservation between the two. Obviously, there's additional complexity with the size of the human protein, but Steve West realized that this was a big deal and invited me to a meeting he was organizing. I literally only just started the group, and that opened so many doors, and I, you know, will be forever grateful for that early opportunity to, as Helen mentioned, go and actually speak at meetings and present the work. And, you know, from that point on, I got into the meiosis field, the genome stability, DNA repair fields, and, you know, have been going and speaking at those meetings ever since. So I think that was really important for me. I'm very fortunate now. My next door neighbour's Peter Radcliffe, and he's my line manager and a bit of a mentor. He always challenges me. He suggests, "Why don't you just give up this DNA repair stuff, Simon? Go away for 3 or 4 months, think up something really new and left field, and I'll support you." And it's having people like that who will challenge you, but also will support you as well. So. Yeah, I very much echo what Helen said. I think, you know, having supporting colleagues is supportive colleagues, and a few of them who really are prepared to put their effort into supporting you. And I'm trying to do that now with the people that I'm mentoring. So yeah, I'm hopefully giving back a little bit.
Lovely, great, thanks, Simon. Let's go on to David next then for the same question. Have there any been particular mentors or collaborators or support networks that have helped you?
Yeah, so I guess thinking back, I mean, certainly when I was a PhD student, I knew I wanted to do science, but weirdly, thinking about it, I never really thought about what the career would look like. I just kind of carried on with the experiments and enjoyed what I was doing. And I remember we were away at a conference, and my PhD supervisor, Steve Busby, said, "You know, you should think about getting a fellowship." I didn't have a clue what he meant, but it's amazing looking back that I wouldn't have known that. But you know, that was the first time that someone kind of steered me in the right direction, and I think Steve has always kind of done that in a very hands-off kind of way, just kind of nudged me in the right direction. Thinking more about the present, I think something I really appreciate almost every day is having good colleagues around me. You know, because sometimes university life can be a bit of a drudge when you're trying to wade your way through paperwork and filling in forms and this system not working and that system not working. But if you've got some people around you who are friendly faces you can talk to, not just about science, but just day-to-day stuff, that's a really big help. And we've got some great people like that in Birmingham that I really appreciate.
Great, thanks, David. And how about for you, Mike? And I suppose the question I'd ask you particularly is, have those sort of mentor and sort of networks kind of changed during your career, or have they sort of stayed pretty
I think
very
it's
much the same?
Yeah, I think it's very dynamic. I mean, you go, you know, your influences, mentors and influences change throughout your career. And you know, during my PhD, actually, I was a very unreliable PhD student until Peter Overath, who was a Max Planck director, came to do a sabbatical in the lab I was working in the Wellcome Labs in Beckenham in Kent in the days. And so I was placed on the same bench as him, and he showed me how to design experiments. And it was like an epiphany. It was like a revelation that this is how you actually got organized. So that was incredibly important influence. In my, George Cross was my first postdoc supervisor. He was fantastic too. Didn't interfere very much, but, you know, always asked the key killer question. Sometimes a slightly deflating question, have you thought about, you know, but fantastic influence. And then in Oxford, actually Alan Williams, I wrote an annual reviews of biochemistry review with him, which was quite a young age for me, not for him. And he taught me a very important thing. He said, we were going through all of this literature, and he said, do you believe in those papers? And I said, well, no, not really. He said, then we don't reference them. And that's something which I've taken with me ever since. If you don't think a piece of science is correct, don't reference it, because it's not doing you or the field any favors. That was quite important. And then in Dundee, Phil Cohen was a fantastic influence on me, showing me that you can dream large. You know, if you want to do something and it's unconventional, don't let that stop you. Just have a crack and do it. So I think those are the individuals that had the biggest influence on me over the years. But yeah, it's always a dynamic, you know, and, you know, working with people like Jeremy Farrar at the Wellcome Trust, you know, it's another fantastic set of experiences, and you learn so much from your peers and friends in science.
Lovely, thank you. I mean, I'm sort of reflecting here on the fact that, you know, the vast majority of these kind of influencers are male. I wonder if any of you have any sort of thoughts about that. Is that just because, you know, science is finally getting to a point where there's more women in positions where they might be able to sort of be mentoring and pushing people on, or is it more complicated than that? Anyone could jump in here, I suppose.
That's a really good observation. I think from my generation, all the senior scientists were male, and it's a terrible excuse, and something that absolutely has to change and is changing, but not as quickly as it should. But I think you made a really important observation, Rachel. Maybe Helen wants to comment.
Yeah.
I would comment that what I find interesting whenever in my school we roll out mentorship programs is how all of the people that ask to be my mentee are all women. And so I think maybe when I was younger, I maybe didn't think it mattered so much that there's role models. But now that I'm at the stage I'm at, I do find, and I would say the majority, I've had some really great male mentors, had some really great female mentors. In the end, it's about the people that are around. And so that's one of the reasons why there has to be an improvement in the numbers of different types of people about.
Absolutely, yes, and diversity more broadly as well, I suppose, wrapped up in that. Okay, great, thank you. I've got half an eye on the time here, so I'm going to move on to a question about obstacles in your career. So what has kind of held you back, and how have you overcome it? And I think, Helen, you sort of spoke to that a little bit in your introduction. I don't know if you want to add anything further to that, or does that sort of encapsulate sort of the main obstacle you've had?
I think just to say that I often worry about this because I'm very open about my background and history, and sometimes I worry about frightening people, because, you know, if you ask most people, do you want to lose your job in the first 8 years of running your lab, they might run away from it. And all I can really say is I'm still here, right? So, you know, it doesn't actually finish you off. And I certainly think at the stage when it happened to me, I think the person that was responsible for the decision had never heard of unconscious bias at that point, for example. And so I'm very hopeful that things are changing with respect to respecting that there are different ways of being a good scientist. It doesn't have to be just one. But I think the biggest obstacles for me in my scientific career is the fact that I'm female. It's as simple as that, and there's not an awful lot I can do to overcome that.
Yeah, okay, yes, I mean, I hear you, Helen. I think we just have to be allies, don't we, to people. I think allyship is a really valuable tool. Right, okay, so who shall I move to next? So David, how about you? What do you think the main obstacles in your career have been?
Yeah, so
and actually, can I interrupt you for a second? I don't yet see any questions in the chat, so just as a reminder to the audience, if you have a question for our amazing panel, please pop it in, because I think we'll be moving over to questions from the audience in about 10 minutes, 5-10 minutes time. Sorry, David, to interrupt. It just occurred to me to ask that question.
Yeah, well, so I was thinking about this. I mean, I don't think there are any specific big obstacles that I can think of, apart from dealing with university admin. But the one thing I would say is that, and I think this applies to pretty much any obstacle, is just when you hit things that are difficult, you've just got to carry on and try again and again. And I did find that difficult when I first started in the lab. So when I first started running my own lab. So when I was a PhD student and a postdoc, looking back, I dealt with failure in quite a rational way. And I always thought of it as being progress. You know, the experiment failed, but we know what to do differently next time. So it wasn't really a failure because we learned something. And then when I started running my own group and, you know, you would apply for a grant and it would get rejected, you know, it would be heartbreaking. And you'd think, you know, what did I do wrong? Why wasn't I good enough? And then you realize it's just the same thing, that, you know, you take that grant, you look at the feedback, you try and improve it, make it better next time. And yeah, so that's what I'd say about obstacles, that lots of small obstacles along the way, but never give up, just keep going.
Yeah, I think so, because like, if you don't apply for a grant, you're definitely not going to get it. So yes, you just have to keep going. I'll move on to Mike next, please, for that same question.
Yeah, thanks. I mean, I've had a pretty charmed career, I have to say. Not too many obstacles, but just the point that David was making. To be a successful scientist, you have to become a tactician as well as a strategist. Strategy is all great, but the art of the possible is really important. And you will have grants turned down, you will have papers rejected, all of this stuff, which is hurtful at the time. But really, you just have to find a way around these little blocks that come along, and be prepared to be agile in your tactics. There's always a third way, you know, and you just need to seek that out, I think. So never let the little setbacks upset you. Just at the moment, the main obstacle I've had in my career is happening right now, which is just trying to close a funding gap on creating facilities so that we don't lose our companies from the place that creates them. And that really drives me mad with all of this sort of stuff about leveling up and so on. But when when a region knows what it actually needs, it's actually sometimes very hard to get that money. So I'm having some political frustrations at the moment in my career, and those have probably been the most intense that I've had.
Yeah, that does sound frustrating. Is that because, you know, they have all these incubator labs around Oxbridge and Imperial that are kind of easy for people to access? Do you think it's sort of facilities driven, or is it people just want to be there?
Yeah, I mean, that's exactly, we just don't have those facilities in Dundee. We're building one now, but we're short of money to complete it. And, right. You know, so we know what we need to do and we're doing it, but actually raising from the public sector a little bit of final money to complete it is proving very difficult. And, you know, when it's so obviously what the region needs to create jobs and keep inward investment in the region, rather than have it bounce off and go to the golden triangle. Yeah. So obvious, but you know, these are the things that try us, right?
Yes. Okay. Yeah, makes sense. Last but not least, to you, same question to you, Simon.
Yeah, I've been very fortunate, but I think part of that has been and Mike's touched on it, and I think Helen and David as well, that, you know, our careers are, there will be challenges along the way, and I think to assume that won't happen, I think, is a little naive. I mean, I think my first major challenge came towards the end of my first postdoc. I'd only been there a short period of time, but for various reasons, one of the senior group leaders in this institute had taken up an executive position at the NCI, and was spending a huge amount of time down there, and there was a mass exodus of postdocs from that lab, and it was almost like, you know, the rats were deserting the ship, and it was quite a bizarre experience, because it went from being 60 or 70 people in a really buzzing, vibrant environment to a few people left over, and that was quite a challenge to accept. And I became disillusioned actually with academia at the time. I started to look for positions else in Boston, in Millennium, for example, and other sort of startup biotechs, pharma, what have you. And I was really lost, I have to say. But I made a decision, and it was a difficult one at the time, to stay in academia, and I moved to another lab very early on. As I say, I'd only been in this group for about 2 years, but I just felt it wasn't right. And to make that decision at that stage, I had an EMBO and the Human Frontiers Fellowship that I took consecutively. That meant a bit of a challenge in terms of continuing. But I just decided that this was the way forward. So I moved institutions, I moved to another lab, and that was a really career-defining moment for me, because if I hadn't done that and hadn't taken this difficult option, I wouldn't be where I am now. And so, although it wasn't an obstacle, so to speak, it was rather a challenging decision to make. And I think we all have them. I mean, we've heard about Helen's story, and how she's grown stronger, I would imagine, as a person, as a scientist. And obviously, David's journey as well. We've heard about his. Did he want to stay in science? Should he become a teacher? They're all difficult decisions to make at the time, but you look back and reflect on where you've been, and there are key moments in your career where you say, okay, God, I'm glad I did that. So I think my message would be, don't shy away from difficult decisions. You're going to have to make them at some point, but be brave, because I think you will look back and be glad that you did them.
Oh, that's a great answer, Simon. Thank you so much. So I've got a couple of questions now that have come from the audience. I think this is a really great question. So as you know, we're sort of celebrating 60 years of the Colworth Medal, and someone in the audience would like to know how the process of being nominated for the Colworth Medal came about in each of your cases. Helen, you sort of mentioned this in your introduction, but so nominations come from the theme panels within the Biochemical Society, but can come from anyone. So yeah, it'd be really interesting to see sort of what happened. And let's start with you, Mike. Can you remember how the process came about for you?
Yeah, I think Phil Cohen nominated me, actually, which was typical of Phil. And actually, Dundee has got an incredibly strong track record of Colworth Medal winners, and it's because we bother to nominate our colleagues that we think highly of. I've personally nominated several people for Colworth, some successful, some not, over the years. So I think some institutes have a culture of nominating for all sorts of things, not least the Colworth, and that's a good thing, but it does mean maybe some people are disadvantaged if they're working in a place which has a culture of not nominating people. So I think that means that the individual committees and so on in the biochem need to be very vigilant about looking for talents. Really, what it should be about is recognizing pure talent. Thanks.
Yeah, absolutely. Sort of more about kind of finding an award for a talented person rather than thinking who to nominate for a Colworth. Is that sort of what you're saying there?
Yeah, I think we need a wider range of people nominating in a way, rather than probably what happens, or certainly has historically happened, of a relatively small number of institutes being particularly good at nominating their own people.
Yeah, absolutely. Okay, and David, how about you?
Yeah, so it was kind of similar. So certainly at the time, we did nominate a lot of our own people in Birmingham for things, and I think I've been nominated for the Microbiology Society Fleming Prize, and someone suggested to me that it would probably be worth asking someone to nominate me for Colworth as well. And I think, Helen, it was the same person that nominated you. I think it was the same person. They warned me. They said, well, you know, I did nominate someone last year as well, so I don't know how that will go again, but it was obviously okay.
Oh, well, that's really good, interesting to hear, isn't it? That there's certain people who are repeat nominators. Yeah, interesting. Okay, and Simon?
Yeah, I think Rachel, you and I discussed it before we came online, and it reflects what Mike was saying. I think certain institutions promote their people, and I don't think that's necessarily fair, because it doesn't cover the full distribution of outstanding people that we have in the UK, right? I mean, particularly for the Colworth. But I was very fortunate, as I mentioned. I was in an environment that was incredibly supportive, and Steve West nominated me for the Colworth, and he was really following in the footsteps of Thomas Lindahl, who nominated Steve for many of his accolades. And Steve has nominated me for every award that I've won since. I'm not sure if anybody else, maybe he's the only person who believes that I do anything of value. Anyway, so you know, that's just the way it is. I don't necessarily agree with it. I mean, you know, we all four of us on here have really benefited from the Colworth, but I think there's probably other people who were equally deserving who maybe didn't get the chance. I think David's point about maybe being bold and suggest, well, why don't you nominate me for this? I don't, you know, it's a little bit of it's a hard one, isn't it, to kind of promote yourself, but sometimes you have to do that in certain environments. And I think if you're in an environment where you don't have colleagues who are historically nominating people, maybe you have to be a little bit more bold to say, you know, would you be willing to nominate me for this? I don't think that's a bad thing at all.
Yeah, well, yes. I mean, I think just on one of those earlier points, of course, for the Biochemical Society Awards, there's a nominator, and then there's 2 letters of recommendation. So there must have been at least 3 people who wrote something positive about you, Simon, not just, you know, yeah. And of course, I think, you know, where all of those nominations, 3 nominations come from one institution, of course, that's not quite so strong as showing you've had impact outside your home territory. Helen, do you want to add anything to your earlier answer? Or
yeah, I would just say that so I nominate all the time. I'm always nominating people from outside the institution, inside the institution, not necessarily just for the Colworth, but I think it's really important to do. And actually, when I was in Dundee, say Nigel Savory, who was a lead of one of the themes, was the person that contacted me to nominate me. Dario Alessi was very happy to support that nomination. So Dundee really did have a strong culture of you know, making sure the people that they thought should be recognised were getting recognised. And I think, you know, it's an active process, right? And it's always very easy for us all to get blinkered and do what we're doing, but some people give a bit more outward-facing work than other people do.
Yeah, and I think it's important to, I can't remember if I've mentioned, I know I mentioned this when we were chatting at the beginning, that the awards committees can only award prizes to people who are nominated. So, you know, that sounds kind of obvious, but, you know, if the selection of candidates is quite small, then, you know, then you're not going to see that diversity perhaps, and if there are kind of more candidates to choose from. Okay, so let's move on to a question sort of more specifically directed at advice for early career researchers. So if you had to give one piece of advice to an early career researcher that might improve their chances of being nominated for or receiving a Colworth Medal themselves, what would it be? Let's start with you, Helen.
Yeah, that's a difficult one, right? I think the only thing I wish I'd known when I was younger is that it's okay to do what I'm interested in doing, right? And actually, I think that's the key. People talk about persistence, but there's survivorship bias in that, right? I think that you have to be really clear with yourself that this is what you want to do, and be prepared for it to all go horribly wrong, but at the risk of not doing it, right? So I think that if you really want to do something, most of us are clever, most of us have good ideas, and we're creative, and I think that people that maybe play it a bit safe and try to follow a track or try to do the things they think they're supposed to do, they stifle their own creativity. And so I think ultimately that would be my advice, is be you. It's okay, right?
Oh, that's a great answer. Plenty of people told me I'd never make a career out of working on borreliosa, because no one's heard of it. But here we go. Okay, so how about you, David? What one piece of advice?
I think I would answer that in pretty much the same way. I think you've just got to do what really gets you excited and stick with it and just go for it. And if you love the science, hopefully, eventually it will love you back, and it'll all work out. But I think there's nothing worse than doing something you don't enjoy or you don't want to do. So I think you've just got to take something that gets you excited, work hard at it, get everyone else excited about it, and then hopefully it'll all pay off.
Yeah, brilliant, great advice. And just a final call for questions, if there's any more from the audience, before I ask for an answer to that question from Mike, please.
Yeah, I would say follow your own. I mean, I think similar to David and Helen's answers, follow your own nose, not other people's. How do you get a Colworth Medal or any kind of award? You make a discovery, and actually the best place to make a discovery is in an understudied area of science. So if you're following the crowd, it's going to be very difficult to make a fundamental discovery. If you tend towards working in the places where are in the too difficult box for most people, there aren't any kits for this, so on and so forth, actually you're much more likely to make a fundamental discovery, and that's really what matters in science in general, and is certainly what tends to get rewarded with awards. So I would always say, you know, seek out the waters less sailed rather than follow the crowds. Yeah.
Oh yeah, wonderful answer. And last but not least, Simon, any thoughts on that?
I think Helen, Mike, and David have expressed it very clearly, but I think in terms of early career, I mean, if we go back to, you know, you're a PhD student, you have the world in front of you, right? And I think I would say that and Helen's probably benefited from this quite a bit, because when you move to new places, although it's a challenge, your growth in learning is almost exponential. You're surrounded by new colleagues who you'll become exposed to new ways of thinking, new technologies, those kind of things. So I think in addition to being true to yourself and working on something, you know, as Mike put it, sort of less well trodden, I suppose, think about you know, where you can be to do that, the different approaches you can apply to that, and you know, being able to travel in academia is one of the real benefits. I mean, moving to another country or another institute in your own country, if you feel that you don't want to leave the UK, for example, that movement is so key. I think is very important for you as a growth as a scientist. I mean, that's really early in your career. You know, from going from a PhD to a postdoc, etc. So I think that for me is going to be very important on top of what everyone else has already said.
Lovely, thanks, Simon. Yeah, really great advice. So we've only really got a short amount of time left. So I'll ask a question from the audience, and I just ask you to keep your answers brief, and please, just with one eye on the time. So how important is choosing the correct PhD, and what is the most important thing to look for when choosing one? And is it the same for a postdoc? So yeah, just any sort of key thoughts on that. Does it really matter where you are, who you work with, what you work on? Is it the subject or the technique in your view? Let's go to Simon first on that one.
I think it's a very difficult thing to know at that stage. You know, I was I had a gut feeling that Cambridge was where I wanted to be with Steve Jackson. He was not known at the time, bizarrely, and it was actually a chance meeting with Gene Beggs in a lift in Edinburgh who said, oh, you should go and do a PhD with him, and that's how I ended up in his lab, right? So thank you, Gene, amazing scientist as well. She's been a great role model to me. But that essentially was it. I was so naive. I had no idea what a good PhD was, and I don't actually think you can know. I just think you need to be in a good environment, surrounded by good people, and make difficult decisions sometimes.
Great, thanks. And Helen?
Yeah, briefly, I had no idea about my PhD other than I wanted to do something to do with structural biology. And when I went and did a postdoc, I went and worked for somebody who'd started their lab 4 months earlier. So how could I possibly have known? I actually just liked the idea of the project, liked the idea of the area. I think being flexible is quite helpful. I think actually drawing out your life in pen at the age of 23, right, is perhaps not the way to find your path.
Very wise, yes. And David, from you?
Yeah, well, I was very much the same, kind of just did something that I thought I'd like to do. I always say to students when they ask about doing a PhD with us or with anyone else is, you know, you could have a completely different experience in one lab compared to the lab next door. So don't just speak to the supervisor, speak to other people in the lab, speak to other people in the same institute, and just go into it with your eyes open is the advice that I would give to people.
Great, thanks, David. And Mike?
Yeah, I think it matters remarkably little actually how you choose your PhD, postdoc, and where you do them. I think there's a bar. You want to be in a very well found place that has good facilities, etc., etc. But nearly everywhere that's research active is that these days by necessity. So that's good. So I think really the most important thing, as David said, is when you go and interview, ask yourself, do the people who work here look happy? Because you know that you're going to spend an enormous amount of your time and in this environment, and if everybody's miserable, you really don't want to be there. So I think this thing about research culture is really important, and you can pick up on what the research culture of a PhD, a lab where you're going to do your PhD or your postdoc is like very quickly by, as Simon says, talking to the other students and postdocs, and just check out their body language, and just don't go somewhere where everybody looks miserable.
Yes, and seek them out if you have the opportunity to be shown around the labs. Ask some questions there. Yeah, absolutely. Well, that's brilliant. Thank you so much. I think I'm going to draw things to a close now. So thanks once again to all of our speakers. You can continue the conversation online. You can follow @BiochemSoc and PP Publishing on Twitter. You can find out more about our awards programme and the latest winners by visiting our website. With the details are on the screen there. Nominations for the 2025 awards will be opening in August this year. And you can find out more information about the webinar series. Propose your own webinar and watch previous recordings on the biochemistry website. www.biochemistry.org/events and training. So please do look on the website for upcoming future webinars. If you've missed any of them, 50+ we've run so far, like to watch them again, you can see them on the Biochemical Society's YouTube channel, and the details of that should be on the screen as well. The recording from today's webinar will also be available to watch in the next couple of weeks. So join the Biochemical Society on the 12th of June at 2 BST for part 2 of this series, when we'll hear from Steve Jackson, who's been mentioned a few times, and today Andrew Sharrocks, Giles Hardingham and Robin May. Finally, I'd like to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. It's an exciting time to join the Biochemical Society's community of researchers and specialists to stay connected and take advantage of key benefits, including discounted registration for society conferences and courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. So please visit the website to find out more details. If there are any PhD students here, the rate for joining for students is very generous. So definitely having a look there. I mean, there are loads of awards for attending conferences, and so yeah, if you're attending a Biochemical Society conference as an early careers researcher and you're a member, then you have a good chance of having some financial support for that. So thank you so much for attending. Thanks once again to the panelists, Simon Boulton, Helen Walden, Mike Ferguson and Dave Grainger. And we'll say goodbye for now. Thanks again.